Have to start because we are short in time. So welcome to everyone to this marine ecosystem session. And just a few reminders to the speakers. So you have a time slot of 10 minutes, but please stay in 8 minutes for the presentation, and then we can save some time for at least one question. If you are running out of time, there will be possibility to discuss during the coffee break, but also during the other session, after the other session, and during the workshop this afternoon, because there is a marine biodiversity workshop. After this session, there will be also a coastal marine biodiversity session that I invite you to attend. So let's start with the first speaker. I invite on here Enrique Montes. Okay, thanks. So the presentation is about scaling image-based marine plankton biodiversity using dynamic satellite seascapes, a contribution of the Southeast US Marine Biodiversity Observation Network. Please, thanks.
Good morning, fellow marine scientists. I'm very happy to see you today. After many talks about being on land, I'm very happy that we're now getting our feet wet. So I'm going to be talking to you about how we're thinking about scaling plankton observations that we collect with imaging devices in South Florida waters as part of the Southeast US Marine Biodiversity Observation Network. It's a program that has been going on for about 10 years in the region. And how we think about scaling point-based observations to larger domains, so we can see how these plankton species show up in different portions of the area. It's a conceptually very simple experiment, but it's very difficult to execute because you need to be able to hop on a ship, go offshore. Being at sea is difficult. Collecting data is not easy. But we're lucky that we are partnering with a NOAA program called the South Florida Ecosystem Restoration Program that has these bi-monthly cruises in the area where we occupy many stations from Biscayne Bay all the way down to the Florida Keys, up on the Gulf of Mexico, the West Florida Shelf, all the way up to Clearwater. So it's a big survey that we complete over the course of 7 days. In these surveys, we collect lots of hydrography, trace metal, primary productivity, carbonate system parameters, but then also lots of biological data, mostly phytoplankton pigments, microscopy, IFCB measurements in coordination with the Florida Wildlife Commission, zooplankton with nettos and imaging, and biooptical properties like absorption coefficients and surface reflectance and so on. So, and environmental DNA data. So the idea is that we can get a very comprehensive picture of biodiversity using multiple tools that we integrate. Now, the question that we're trying to answer here is to what extent we can resolve biogeography and phenology of different plankton groups in the area using satellite data. This is a big question for us. How are we collecting the plankton? We can either collect plankton using nettos, but that's very laborious. You need to drag a net, then you need to process the samples in the lab with a microscopy with an expert in taxonomy that can look at these organisms, identify them, and that takes a really long time. So we're taking a different approach. We're using cutting-edge technologies. We're using a continuous particle imaging and classification system, which is basically an underwater microscope that uses dark field illumination to capture images of the plankton in vivo. And the way we do this is we attach the instrument to the CTD rosette, and in every station where we do a CTD cast, we also get a profile of the plankton. And this is more or less the kind of images that you can get with this instrument. In the top, you will have different types of chain diatoms, Chaetoceras, other species, but also single-cell diatoms, trichodesmium, some dinoflagellates as well. Things that are in the range of a few tenths of microns to a few millimeters. So it's the larger fraction, right? And also many species of zooplankton that show up in some of the stations, and then they don't show up in other stations and in other portions of the year. With these measurements, we're starting to get a sense of how these different phytoplankton groups are distributed throughout the survey, and we're finding a few hotspots, especially in the coastal zone, right near areas where there is a lot of river discharge, like in the Caloosahatchee River. It's a river that connects that big lake, the Okeechobee Lake in Florida, that drains onto the West Florida shelf. That's a place where we see major, very thick blooms of chain diatoms, of mixed communities, but also we see single species blooms like Skeletonema showing up in these areas. But we see Trichodesmium, which is a nitrogen-fixing phytoplankton, and some species of dinoflagellates. We also see very dominant groups of zooplankton like copepods are coinciding in these areas where there are, these phytoplankton blooms are occurring very near the coast. They tend to dissipate as you move offshore. Larvaceans, which are filter feeders, echinoderms, and then interestingly, we see ostracods that they tend to prefer more offshore waters. So we get a spatial picture of this, and we also we're starting to get a sense of the seasonal or temporal changes in these communities. With 2 years worth of data, we're somewhat confident that we're seeing something that looks like a seasonal pattern in the diatom blooms, but also the copepods and other groups where their concentrations tend to be lower during the winter and springtime, and then they go up in the summer and fall, and then they come back down. It does not include data from January this year, but I can promise you that those values are going down again in the winter. Now, these are, of course, point-based observations, single locations, and the question is, how do you scale these using satellite data? And for that, we're taking advantage of a very cool a product that my colleague Maria Kavanaugh from Oregon State University developed some years ago. It's dynamic satellite seascapes. It's a very useful model in that it integrates multiple types of ocean color data like chlorophyll, CDOM, fluorescence, with physical properties from satellites and from model outputs like sea surface temperature, salinity, winds, integrates this through a machine learning algorithm that uses self-organizing maps and hierarchical classification to render biogeographic units that you see on that map on the left. Each color is representing dynamic surface biome that expands and contracts over time. And for all of us that work in the ocean, we know that this is really important because unlike land, where a forest that is showing up in January will very likely be exactly the same the next January, in the ocean, these habitats are very dynamic and change in time. So, the seascapes have 8-day and monthly integrations at 5 kilometer pixel resolution. It's a global classification with 33 classes, including sea ice. So what we're doing is that we're matching these plankton observations that we do during these cruises with a corresponding seascape category that we detect for the week of that cruise. And this is how the seascape looks like for September of last year. We're seeing that there are 7 dominant classes that are showing up in the area. Some rare classes show up occasionally, but it's very hard to detect them. And we're taking advantage of all the data we collect during these cruises, and we certainly detect or observe that different seascape classes contain different biogeochemical properties. For example, the warm blooms high nutrients class, which is the yellow one that you see there, tends to have much higher chlorophyll concentration and nutrients as well. Whereas the tropical seas, which is the green one, the light green one that you see there, they tends to have lower chlorophyll, lower nutrients, and higher salinity. So we do confirm that these satellite-based biogeographic units are different in their biogeochemical makeup, right? The seascape makeup of the area changes drastically in a seasonal basis. These are, you can see how they change over seasonal cycles, but also from one year to the other. we see that the two or three dominant classes that you see there alternate in a seasonal fashion. And what we're seeing that, for example, towards the end of this year, we're seeing the higher dominance of tropical, subtropical upwelling class, which is a class that doesn't show up very often, but this year is appearing.
2 minutes left.
Okay, I think I got a couple more slides. We're matching then the plankton observations that we detect in each of these classes. Certainly, we see differences in their composition and the relative composition of different groups, but also the concentrations of the phytoplankton and the zooplankton may vary a lot between one class and the other, right? So, these allow us to confirm that tropical seas is a is a class that contains many species of phytoplankton. It's high pteropod concentrations, whereas the warm blooms is where we see the big, thick diatom blooms. Diverse zooplankton as well showing up in those areas. So we're starting to get a sense of how the seascapes can be used as indicators for plankton biodiversity. And I'm going to leave you here with some future work that we're planning to do. We're planning to integrate CPEX data with IFCB observations that are also taking place during these cruises. This is a major challenge. We also collect lots of eDNA data that we're trying to match to imaging observations. And we're thinking about developing indicators based on seascapes that can help, for example, the natural marine sanctuaries inform the state of their ecosystem, right? And there are some recommendations here that I'll leave you to it so we can wrap up. Thank you so much.
Thanks a lot. We have time for a very quick question. So, there are no questions? Okay, the time is run out. Thanks a lot.
Thank you. And I will now call to the stage our second speaker of the day, Riccardo Nanni, please, from ISMAR, the Istituto di Scienze Marine from the CNR. And he will be talking about Mediterranean 4D seascape based on phytoplankton phenology detected from satellite observations, patterns and drivers. Please.
Thank you. Today I'll be presenting the main results of a study conducted in the framework of 4D MedSea project. The ocean, though seemingly uniform from above, hosts diverse physical and biological processes. Regionalization group areas with similar biogeochemical properties, unveiling unique regional characteristics. A well-known example is Longar's regionalization, which divides the global sea in 27 distinct provinces. A common method for defining bioregions analyzes phytoplankton phenology using satellite-derived chlorophyll A data. By tracking seasonal events like the initiation, peak, and end of phytoplankton bloom, is it possible to clusterize areas with similar phenological patterns? Bioregionalization is crucial for studying climate change impacts, as shifts in phytoplankton dynamics serve as early indicators. It also supports sustainable fisheries management by informing ecosystem-based strategies. However, resolution matters. While global classification provides finer details for While global classification provides a broad framework, regional studies reveal finer details. For instance, the Mediterranean Sea, classified as a single province in Longar's scheme, exhibits distinct bioregions at a finer scale. In fact, in 2009, the Hortensio team classified the Mediterranean Sea into 7 different clusters. By using a 10-year daily datasets at 18 kilometers spatial resolution, they generated climatological annual chlorophyll cycles for each pixel and grouped them by seasonal patterns. This analysis identified 2 coastal clusters, one orange and one red, one bloom-like cluster, yellow, one intermittent cluster, green, and 3 no-bloom clusters, cyan, blue, and purple. However, some limitations exist. And low spatial temporal resolution introduced high level of noise and excluded many coastal areas, while the climatological clustering overlooked interannual variability, and surface chlorophyll-based classification did not account for depth-related bioregionalization. Taking inspiration from both the insights and limitation of this Mediterranean study, we defined 3 key objectives. First, we aim to update Mediterranean bioregionalization by extending the time window and improving resolution. This is achieved using Copernicus products datasets, which combine data from multiple satellite sensors along with a region-specific algorithm to reduce uncertainty in chlorophyll measurements. Second, we explore interannual variability through annual clusterization, identifying stable and variable areas and linking them to physical drivers. Third, we extend clusterization to deeper layers using the 4DMed dataset, which provides a 4-dimensional view of chlorophyll dynamics from 2016 to 2021. Overall, the updated bioregionalization largely aligns with the findings of D'Ortensio, confirming the general positioning of the 7 cluster. However, the improved dataset significantly reduces noise and provides a more precise definition of cluster boundaries. The most notable changes appear in the coastal regions, where the red cluster is now more tightly associated with specific coastal areas, and orange cluster extending around it. In the Levantine Sea, the green intermittent cluster has expanded, now incorporating also 2 areas of yellow-bloom cluster. The 3 no-bloom cluster exhibits similar seasonal cycles, following a smooth trajectory. The green cluster has a narrow peak with a gradual increase and a steep decline, while the yellow cluster shows an even sharper peak, characteristic of bloom events. Coastal clusters peak earlier. The orange cluster has a steep rise followed by a gradual decline, whereas the red cluster follows a completely different pattern from the others. These 2 coastal clusters also show the greatest divergence from D'Ortensio cluster cycles, mainly due to the inclusion of newly considered coastal areas. To analyze interannual variability, we applied clusterization to each individual year, identifying 26 different cluster maps. By calculating the frequency of the most prevalent cluster for each pixel, we produced a coherence map. While overall coherence exceeds 50%, the map highlights key areas of stability in red and variability in blue. The most stable regions includes the Gulf of Lion, corresponding to yellow-bloom cluster, the Gulf of Gebes, corresponding to red coastal clusters, and a curved area between Greece and Western Turkey, corresponding to blue no-bloom clusters. Conversely, the most variable regions, such as the Adriatic Sea, southern Sardinia, southern Adriatic, southern Ionian, Levantine Sea, and Aegean Sea, corresponds to the green intermittent cluster. To identify the physical drivers of change, we conducted a preliminary analysis using Sea Surface 8 data. SSH provides indirect insights into ocean currents and helps detect circular gyre structures. We computed a climatological map to visually identify correspondences with bioregional patterns. Our findings show that highly coherent areas of yellow cluster align with major upwelling zones, while circular variability hotspots correspond to the key gyre in the southern Adriatic and Levantine Sea. For the final objective of our study, we used the 4DMed Sea dataset, which provide 3-dimensional chlorophyll values derived from a neural network. This daily dataset, with a 4-kilometer spatial resolution, spans 6 years and includes 148 depth layers from 3 meters to 150 meters. To incorporate depth, we integrated vertical chlorophyll profiles for each pixel into a 2-dimensional array, which we then clustered. The resulting map showed notable difference from surface-based clustering, particularly with an expanded presence of the coastal clusters and a distinct redistribution of the three non-bloom clusters. However, the green and yellow cluster in the Gulf of Leon remained largely unchanged, likely due to a strong surface chlorophyll signal dominating the overall vertical profile. In summary, our results align with Hortensio's bioregionalization, but refine cluster definitions, especially in coastal areas. Coherence analysis revealed both stable and variable regions, with circulation patterns and gyres likely driving interannual variability. Depth-based clustering showed significant differences from surface patterns, emphasizing the need for a tridimensional approach. Further research is essential to better understand biophysical interactions and connect surface phenology with deeper ocean dynamics. Thanks for your attention.
Thank you, Ricardo. Perfectly on time. So if you have any question, please. I know it's still early in the morning, but yes.
Great work, wonderful. I'm wondering if you can use this approach to get a sense of how these different categories that you're aggregating can be examined on a monthly basis through time, through the satellite record, if you're thinking about doing that.
Yeah, with this approach, is it possible to analyze monthly changes, but not doing a clusterization because clusterization is based on the annual cycle of chlorophyll. So not every area of the Mediterranean Sea has a bloom in the same period, so it is essential to have an annual cycles to distinguish the areas who blooms earlier and the ones that blooms later.
So this is more working like a climatology. Okay.
Thank you.
Ricardo, right?
Yeah.
Yeah, thank you, Ricardo. Very nice presentation. I was wondering if you did or you plan to integrate PFTs, like in your you know what I mean? When you look at the phytoplankton groups as part of the satellite-derived product, to understand in your phenology, what are the group that are the dominant ones at the phenological cycle?
Yeah, we thought about that, and we are planning to include it in the further analysis that we want to do, because we want to have a broader visual of the dynamics of phytoplankton, so we want to include also PFTs.
Yeah, that'll be cool. Yeah, to see which ones are changing, right, in the long time series.
Yeah.
Yeah. Thank you.
Okay. Thank you, Ricardo. So we move to the next speaker. The next speaker is Maria Laura Zoffoli from CNR ISMAR. And she's going to speak about our absorption diversity of bloom-forming phytoplankton species toward epispectral remote sense identification of red tide events.
Okay.
The floor is yours. Thanks.
So good morning, everyone. I'm here to present on behalf of Pierre Charnès, who is the first author of this work. I don't know if it comes. Okay. So here we are. So the presentation is about the possibility or the potentiality of hyperspectral data to discriminate in between different phytoplankton bloom forming species. So red dyes are accumulation of very concentrated patches of phytoplankton. Some of them are formed by species that potentially can produce toxins, and others, just the accumulation of cells can produce imbalances in the ecosystem, for example, with hypoxia events, which brings to mortality of fauna, and also with losses, economical losses, and ecological problems. They are very frequent in coastal areas, mainly due to eutrophication, and they are expected to increase in the upcoming decades. So they are a big concern worldwide. Traditional monitoring programs are based on in situ samples, but these events usually are very fast, very dynamic, very patchy also, so sometimes the in situ sampling loses the occurrence of some events. So satellite remote sensing brings a potential tool to evaluate the dynamics, to map the extension of these hubs, and also eventually to identify other occurrence. So the question is what we can identify eventually in terms of species with remote sensing, hyperspectral mainly. So today there are 269 species identified by the UNESCO as harmful algae blooms, as forming harmful algae blooms. From them, 215 potentially can produce toxins, and the others only brings high accumulation and imbalances to the ecosystem. But from all the groups, the more representative species are dinoflagellates. So today there are some groups that can be identified. For example, cyanobacteria can be eventually identified by multispectral remote sensing. Some species like Noctiluca or Mesodinium or the cryptophyte that they feed on, or some waters dominated by Lepidodinium chloroforum. Eventually they have different pigment composition and they can be identified by multispectral remote sensing, but other classes are much more challenging because they share the same pigment composition. So in this work we are focusing on those most challenges species. So the questions that we have is it possible, for example, to discriminate in between a dinoflagellate that bears peridinin from another bearing fucoxanthin, or eventually can we discriminate from diatoms from another bloom formed by rhapidophytes or aptophytes or other dinoflagellates? So to answer this, we worked with monospecific phytoplankton cultures in the lab. We estimate the particulate absorption coefficient either with APSICAM or measuring on the filter pads the absorption. As a total, we use 60 species and 164 hyperspectral spectra. And some of the species we compiled from the literature, and other we perform in the lab new cultures. So we filter first this absorption coefficient. We calculate the second derivative, and we perform a hierarchical cluster analysis. So here we are. From these most challenging species that we focus on, we could identify 4 main groups. A main group formed by aptophytes, except Primnesium parvum that didn't classify well. A second group represented by diatoms and rhapidophytes. A third group represented by dinoflagellates with fucoxanthin, and in this group it was incorrectly classified also Primnesium parvum, that is an aptophyte, and finally a fourth group represented by dinoflagellates with peridinin. So we knew from previous works that some other groups can be easily identified as cyanobacteria, cryptophytes, prasinophytes. And in this work, we focus on the most challenges species. We demonstrate that hyperspectral data indeed can discriminate in the absorption spectra from different groups. So with some exceptions, that one species that didn't classify well, we can identify aptophytes, we can identify dinoflagellates either with the peridinin or fucoxanthin, and we can identify diatoms. So the next steps, here we focus on phytoplankton and absorption. So the next step is moving forward to reflectance. So what happens with reflectance indeed? So first, using in situ data acquired over concentrated blooms or extracted from satellite hyperspectral images. There is a poster we are going to present tomorrow about this. And finally, using simulated spectra with the transference, radiative transference to see the impact of the different absorptions on the reflectance spectrum. So that's all.
Thank you, Laura. We have time for questions. Yes, Victor?
That's very nice evidence. A quick question: how about the backscattering? Are you also trying to, when you do the radiative transfer simulations, are you thinking? Do you have any measurements of?
Yes,
lab,
we have measurements. I mean, the ones that we did in the lab, we have measurements of backscattering. So we are planning to include in this work. We use only absorption, so there is no backscattering considered. But probably backscattering can be another factor to differentiate in between different sizes of cells eventually, yes.
That will help very much, yes.
Yeah.
Nice. Very nice work.
Thank you.
Yes, Peter?
Hi, Peter Miller from PML. Yeah, looks really interesting in situ results, but I wasn't sure if you evaluated how well you can distinguish between those four species.
Between those species, I mean, the cluster, we use a method, an objective method, where it gets to a plateau or something like this, where we should stop cutting the groups. So these are the groups that we eventually can identify. So I don't think so far in this analysis we couldn't see nothing further in terms of species. I mean, so far groups.
Okay, and you think you'll be able to distinguish those groups from PACE, from the satellite data?
So in some cases we are working here in very concentrated blooms and almost monospecifics, because they form like almost monospecific events. So eventually, in this case, we can differentiate one group to another from remote sensing. Depends on the groups indeed.
That's very nice. Thank you.
Thank you.
Thank you very much, Laura. We'll move to the next speaker. And we'll welcome Maria Ruao Lima from CIMA, the Center for Marine and Environmental Research in Portugal. And Maria will talk about phytoplankton assemblage structure of southwestern Iberia, combining complementary approaches to assess variability and underlying drivers. Please, Maria.
Thank you very much. So good morning, everyone. Today I'm going to share with you some of the insights from a past project on the variability patterns and the driving forces of the phytoplankton assemblage structure of Southwest Siberia using complementary approaches. So why study phytoplankton of Southwest Siberia? The Phytoplankton are key primary producers, regulators of ecosystem functioning and services, and are considered relevant indicators of the environmental status. Of course, the different functional traits and niche preferences lead to the different responses to the environmental conditions. And this area is characterized by high levels of mesoscale variability and was classified as being significantly vulnerable to climate change. The current challenges that we have are the limited knowledge on phytoplankton structure of Southwest Siberia, the lack of a comprehensive understanding of the environmental determinants that shape the assemblage structure, and then most satellite algorithms are global and perform poorly at complex coastal waters in deriving the phytoplankton composition from space. So the study objectives were to assess the spatial temporal variability patterns of the phytoplankton assemblages of Southwest Siberia, identify the underlying drivers and predictors, and evaluate the performance of satellite algorithms at a regional scale. The field sampling was conducted under a past project named Phytoclima that included regular sampling campaigns along 3 stations, F1, F2, and F3, as you can see in the map, along a coastal offshore transect and a 2-year period. The sampling was performed at 3 depth levels that are representative of the first optical depth in the water column, and the sampling time was within a 3-hour window of the Maudis Aqua passage. We have also considered additional samples from an oceanographic cruise during summer 2013, but only to increase the number of samples considered for satellite algorithm validation. So a comprehensive set of variables was considered, including physicochemical variables as well as phytoplankton-related variables, and these were retrieved from different sources, including in situ observations, remote sensing, and models. And then what we did was to try to relate the environmental conditions to the variability of the assemblage structure. We applied multivariate analysis, such as non-metric multidimensional scaling and canonical correspondence analysis. We have also estimated the chlorophyll contribution of different size classes and functional types from the abundance in biomass obtained from microscopy techniques, specific diagnostic pigment analysis fine-tuned to the North Atlantic, and using a recent approach of chemical taxonomy analysis. We have then compared the results from the diagnostic pigment analysis with satellite-based algorithms, either using a spectral-based approach that basically links the variances along the reflectance spectra with the different size classes and groups using empirical orthogonal functions, and an abundance-based model that estimates the contribution of different size classes as a function of total chlorophyll A, but with a twist here, so considering the influence of sea surface temperature on the model parameters. So the satellite products that we considered were all multi-sensor and were retrieved from the Copernicus Marine Service platform with a daily resolution and over the 2-year period. And the differences between them were in their processing frameworks and providers, the spatial extent and resolution, and the available variables. So 2 of them had both size classes and functional types, and 1 of them only size classes. So moving on to results, the microscopy results show that overall high biomass coincides with a diatom dominance, showed in blue, but there were also different patterns among stations and seasons. We see that in the coastal station F1, there's a dominance of diatoms and dinoflagellates, whereas at the furthest stations, the cyanobacteria surpass the dinoflagellates contribution. We have also seen that there is a diatom increase in winter, early spring, and then an increase of cyanobacteria and dinoflagellates in summer and early autumn. We have also detected a dinoflagellate bloom represented by the green peak in the graph and the arrow, which corresponds to a dinoflagellate bloom in summer 2013 that was coincident with an upwelling relaxation event that was followed by an upwelling pulse that transported the cells from the coast towards offshore. These results were also confirmed from the HPLC, which showed an overall dominance of the fucoxanthine pigment associated to diatoms, and also an increase in the peridinin accessory pigment of dinoflagellates, showed in green, around summer 2013 as well. So the ChemTax results show that overall the groups that contribute the most to total chlorophyll A are diatoms, prasinophysians, and dinoflagellates, although there are some other groups that are also relevant in other stations, such as haptophytes, represented by the dark purple in station F2, and the premonizophytes in light purple in station F3. Then we compare the results with the ChemTax results with in situ biomass, and they overall agreed well, but the best results were obtained for cryptophytes with a higher correlation coefficient and lower errors, followed by cyanobacteria type 2. These different patterns between stations and seasons were also confirmed by the ordination plot, where we can see that F1 samples appear clustered on the left plot area and F2 and F3 on the right plot area. The canonical correspondence analysis identified a total of 3 environmental variables. They are the best predictors of the variability patterns in the assemblage structure, including the wind-derived upwelling index, nitrite, and suspended particulate matter concentrations. So from the satellite algorithms, the comparison with the diagnostic pigment analysis, we saw that overall, looking at the bias column, the satellite products tend to overestimate the phycophytoplankton fraction, the corresponding prokaryotes group, and the dinoflagellates, and this is probably because prokaryotes and the phycophytoplankton fraction are more abundant in oligotrophic areas where the satellite signals are weaker, and hence it's harder to detect them accurately, and the dinoflagellates produce also weaker optical signals due to their smaller sized cells. They are likely influenced by other inherent optical constituents. But we also know that the dinoflagellates group specifically can also be underestimated from pigment analysis due to the ambiguity in the pigment composition. We also saw that the abundance-based model that accounts with the influence of SST in model parameters was best at deriving the nanophytoplankton fraction as well as diatom and dinoflagellates, whereas the spectral-based algorithms are better at retrieving the phycophytoplankton fraction and the corresponding prokaryotes group. For the microphytoplankton fraction, we got similar results from the spectral and the abundance-based model. So this basically tells us that including an additional environmental variable gives us some extra information about the spatial distribution of the groups and size classes that the multispectral remote sensing reflectance data cannot give alone. So to conclude, diatoms and dinoflagellates dominated at coastal stations, while cyanobacteria surpassed the dinoflagellate contribution at the furthest stations. There was a diatom dominance in winter, spring, cyanobacteria and dinoflagellate dominance in summer and autumn. The upwelling index, nitrite, and suspended particulate matter concentrations were the best predictors of the variability in the phytoplankton assemblage structure, and the satellite algorithms need further refinement and validation to be accurately applied at regional scales. So the next steps will include trying to estimate nitrite concentrations from empirical regressions, considering in situ or satellite-derived water temperature, silicate, and chlorophyll concentrations, because our results were not really reliable. There were some errors in the processing. So we tried to correlate with the models, but they did not correlate well, so we will try to estimate this and then apply the multivariate analysis with this model-derived nitrate concentrations. We'll also explore specific plume events, such as the one that occurred in summer 2013. And I leave you some of the further recommendations to wrap up. Thank you very much. Thank you.
There is time for one question. Yes, Maisira, please.
Hi, thank you. So it's Maisira Costa from Canada, from the University of Victoria. So thank you for the presentation. I was wondering, because we had to look at this issue before, that's why I'm asking you. Did you look at how homogeneous is the spatial distribution of your phytoplankton groups in relationship to the pixel size of the Copernicus database that you use for retrieval of PFTs?
So we actually used like a 3x3 grid Yeah.
That makes, is it a 4 kilometers in a 3x3 grid, or was it?
It depends on the product. Could be 1 or 4.
Yeah. So did you use the 1 or the 4? I mean, regardless.
Both. Both.
Okay. So and then so
and
when you look
depending
at,
on the group and size class, I got better results.
Yeah.
With the 1 or the 4, depending.
because because one thing that we encountered before is that for the PFTs, like when we're in the middle of the Pacific, where things are a little bit more homogeneous, spatially speaking, the reflectance can be representative of different for the UF model, right? Because we use similar as the model that you use. And then when you start to get close to the coast, where spatially it's much more heterogeneous, right? You have the mix of different groups, so it gets really, oh, that's a mess. So I was wondering if you had the opportunity to look into that. What is the spatial variability?
I would say from knowledge that we have, even from past projects, that it can be quite different.
Yeah.
The southwest coast, from the east near the Guadiana, and the very center near Cape Santa Maria. So it can be quite different.
Yeah.
I will next analyze the results from the Marbis campaign. So we have, I don't know if you saw the little black squares in the map. So we have stations all along the southern Portuguese coast, and we will try to analyze if it's effectively different or not.
Yeah. Okay,
Yeah.
cool. Thank you.
Thank you very much.
Thanks a lot, Maria.
Thank you.
So next speaker is Roy El-Ourani from Laboratoire d'Océanographie et des Géosciences, France. And he's talking about linking satellite to genes to observe the phytoplankton community structure from space.
Yes, thank you.
Thanks.
So hello everyone. I'll be presenting this work that have been done collaboratively with all of these my co-authors. So I acknowledge them and I acknowledge all the institutions that have helped us to do it. So what is a community structure for phytoplankton? So it's not moving. Sorry. Okay. So, yes. So what is a community structure? In fact, we have different categorization of the definition of a structure, and what we see mainly in the main categorization that we can see is taxonomic groups such as diatoms, dinoflagellates, etc., or functions, so silicifiers, calcifiers, and as well size ranges, the smallest to the largest. But in the ecosystem, these categorization can overlap. So we have to define what is exactly a community structure that we want to study. So in my work, we deal with the community structure as taxonomic groups, so diatoms, dinoflagellates, etc. When we look on the advancement the works that have been done on the community structure from space, we all see that it's all based on pigment compositions and HPLC measurements, since we have a huge data set of these measurements on coastal and oceanographic and bioregions all over the globe. But how does this work? In fact, we measure a pigment and then we associate certain pigments to groups such as fuchsanthin for diatoms, pyridinin for dinoflagellates. And why we do this? Because it's a methodology that covers at the same time prokaryotes and eukaryotes alike. But there is a problem with this, is that it gives a biased information on what is there in the water. Because in groups we have overlapping of phytoplankton pigments, and we have very large uncertainties in precising what is there. For example, dinoflagellates or haptophytes having the same pigment as diatoms. So is there any alternative methodology that can reduce this bias? So this is where we lead to the work in Terra Oceans, where they have identified a gene that encodes a protein. It's called PSBO. What is a PSBO protein? It's core subunit of the photosystem 2, which is a subunit in the chlorophyll, unique to organisms that carry out oxygenic photosynthesis. And when we look at the PSBO gene, it's a single copy gene. That means then when we count the quantity of PSBO, it's a proxy of cell abundance. So each time we count PSBO, it's like counting one cell. So this methodology have been applied on the Tara Ocean expedition from 2009 to 2013 on 145 observations. And then a database has been deduced. The first one is the relative cell abundance on 8 phytoplankton groups, 7 defined as diatom, dinoflagellate, lean algae, cryptophytes, haptophytes, pelagophytes, and prokaryotes, and a non-classified group which is majorly accounting to 5% of the total cell abundance. And from this data set, we have derived what we call the chlorophyll A fraction for each phytoplankton group. And how? Because we integrated the size fractions that have been measured on board of Tara Ocean, which we have 4 sizes from the smallest to the largest. So now, moving out to the cuisine. So how does this algorithm work? So we tried to statistically model the phytoplankton community structure from satellite data. So we have 7 phytoplankton groups expressed as chlorophyll A fraction and relative cell abundance, plus the unclassified group. We matched this observation with satellite data, such as reflectance at several wavelengths, chlorophyll A, and as well physical and bio-optical context measurements, such as sea surface temperature, PAR, fluorescence in the red, and attenuation coefficient. We have 2 major problems in this. The first is we have a very low number of observations with very high noisy data. And we have another problem, is the multivariate aspect of this problem, which is we want to, from one set of predictor, predict a set of a community structure. So I want to give, for my model, one observation from satellite to give me all the community structure at once to conserve the intervariation between phytoplankton groups. So we have used what we call the self-organizing map classification procedure, which is an unsupervised neural network classification method that allows the projection of high-dimensional database into a 2D discrete map. And it's very efficient, so this is where we address the problem within the data. It's very efficient to interpolate and reduce the noise within data sets because of the topology, the intercorrelation between variables. So from this, we have derived maps of relative self-abundance of different phytoplankton groups with their chlorophyll A fraction and uncertainties related to different possibilities of community structure from one set of predictor, because we have very high variation within the community structure is quite complex to address all at the same time. So we have derived the uncertainties of it. So these are the plots for the cross-validation and the test. And we can see that it performs well with the chlorophyll A fraction on majorly all the phytoplankton groups, and as well for the relative cell abundance. And all of this information that we can get, we try to summarize them into bioregionalization, and this is our community structure based on a regionalization, and this is where we get to this type of results, where we have majorly 5 community structures that come out from both relative cell abundance and chlorophyll A fraction information, and we can track them on the as a dominant community structure, as you can see on top, and track them with time in terms of latitude in this case. So each community structure has its own variability within the phytoplankton groups in terms of cell relative abundance and chlorophyll A fraction. So to conclude, so using genomic database such as PSPO or other markers can be a step towards a better definition of phytoplankton groups from space. There is more omics data that are coming soon, like noting the Tara Pacific and the Tara Europa expeditions, and that they will increase the input into PSPO dataset and other genomic dataset. And from this satellite-based phytoplankton community, we can now address more ecological questions, such as the testing of biodiversity and ecosystem functioning relationships and hypotheses on a global scale within time and space. As you can see here, we compare the Shannon index, so the diversity of different relative cell abundance groups versus the primer production, and you can see that this relationship changed throughout space and time. As you can see, space on the from left to right, and time from up to down. So as a recommendation of this work, it's important to integrate and harness cross-disciplinary expertise throughout machine learning, ecology, and remote sensing to push forward collaboration to better observe diversity from space. Machine learning can help and can solve nonlinear and intricate problems such as the homogenization of multi-source data to extract common patterns between remote sensing and ground truth. And we should include global biodiversity maps as this and EBVs linked to ocean ecosystems. Thank you.
Thanks a lot. So, Victor, there is one question.
Very nice work, thank you. A very quick question. You say that this PSBO marker is related to relative abundance.
Yes.
And you compute it as a fraction of the chlorophyll.
Yeah.
Do you think, or is it not reasonable, I don't know enough about that, but it would be reasonable to try to convert that into carbon? And would it be possible? And what would you need to do that? Or it is just a stupid idea and then it's okay?
Yes, I can say that there is a group in Genoscope doing this, like calculating from since here we're dealing with relative cell abundance, they developed a method based on PSBO to give us the real count of cells that has been validated to microscopy and cytometry, etc., from these groups. Then from this abundance, like the real abundance of cells, they calculate the carbon. But it's a large, there is a high uncertainty in the estimations, and we're trying to deal with like this noisy information at this point of the research. It's doable. Okay.
Okay, thank you very much.
Thank you.
The next talk will be given by Pedro Junger from Institut de Biologie de l'École Normale Supérieure in Paris, France. And Pedro will talk about the satellite genomics approach to explore phytoplankton iron ecophysiology in the global ocean. Thank you, Pedro.
So, good morning, everyone. Thank you, Magellan, for organizing this section and everyone for listening to parts of the research that I have been conducting in the bunds. And now I will move from community structure to phytoplankton ecophysiology by bringing together satellites and genomic data to explore it at a global scale. So it goes without saying here that phytoplankton is very important because it's contributing to almost 50% of prime productivity on Earth, but this prime productivity can be limited by different resources, including nutrients such as iron. So iron is limiting up to 40% of prime productivity of the ocean surface. And however, phytoplankton have developed molecular mechanisms to deal with iron deficiency. And here you can see in this simplified scheme of the photosystem that they can replace iron-containing proteins and by iron-free counterparts. So here you can see ferredoxin and cytochrome C6 as iron-containing proteins being replaced by flavodoxin and plastocyanin, right? So here you can define, based on this knowledge, two iron status index for phytoplankton, the flavodoxin index and the plastocyanin index. So what we did is we built an extensive reference database of these genes and transcripts, and we mapped it in the Tara Ocean's metagenomes and metatranscriptomes. And then we got these kind of results where you have the genes and transcripts relative abundance correlating negatively with iron concentration from biogeochemical models such as PIS2. So So here we can see that we have got good correlations between this index and iron in different size fractions of the Tara Oceans data sets for meta-G and meta-T data. So next, we also extracted the satellite matchups for the fluorescence quantum yield, which is a classic remote sensing metric for iron for phytoplankton stress in the ocean. And here you can see that we found first negative correlation with the matchups for iron concentration for the Tara Oceans data sets, and then we found very good correlations between this index and the FISAT metric, which is probably good news. And what we did here as an exercise was training a very simple random forest model based on this Tara Oceans training data set, using as predictors the satellite variables such as sea surface temperature, chlorophyll A, EPAR, and fluorescence in the red, as well as nutrient concentrations from the World Ocean Atlas and from biogeochemical models to generate this kind of biogeography of these iron stress genes and transcripts, and then we validated it quantitatively and qualitatively by comparing with different methods. I'm going to show you in the next slides. So first, here are the results for the flavodoxin index, which are coherent with the current state-of-the-art knowledge in biochemical models. And we've got like variance explained of around 60%. And here you can see on the bottom, we use the leave one out cross-validation as a way of validating internally the model. And as you can see, we have good correlation between in situ and estimated values for the meta-G. For meta-T, we have more uncertainty for the low estimated values. And then for the plasocyanin index, we have a lower performance, let's say, with lower explanation, but it's still with relatively good error rates considering the training data set. And here, the good news is a way of validating the methods, we obtained the predicted flavodoxin index for corresponding to the geolocations where people conducted bottle experiments in the ocean. And here you can see that we got a higher predicted flavodoxin index in the stations which are very far, by the way, from the Tara Ocean stations that have iron as the primary limiting nutrient. And the same pattern was observed for the plasocyanin here on the right, on the left. So these are the meta-T data, so these are transcripts, so these are expressed genes. However, when we look at the patterns with the meta-G data, so the presence of the genes, we do not have the same response, which indicates that, in fact, the meta-T based index are a better proxy of iron deficiency in the ocean. So then we mapped the same reference database in the Tara Pacific data. And we found again now good correlations not only with satellite biogeochemical model products, but with real in situ iron concentration measurements. So here you can see that flavodoxin index and plasocyanin index are negatively correlating with in situ iron concentrations for the different size fractions of Tara, and for meta-G and meta-T with a little bit more stronger correlations for the metatranscriptomic data. So to sum up my presentation, so we did find patterns of genes and transcripts abundance, which are consistent so far with biogeochemical models, with iron concentration measurements, and remote sensing. Which indicates that these genomic proxies can work as a good proxy for iron limitation in the ocean. So then we use the prevalence of these genes and transcripts involved in iron response to develop this kind of of maps of inferred nutrient stress, and we found that meta-T predictions were more coherent with the bottle experiments, and that's very important. We also found that the meta-T correlated better with the iron in situ measurements in Pacific, and so it shows that this workflow provides a good foundation for linking genomic and remote sensing data, but we highlight again the importance of having more in situ data, and that's one of the perspectives of this work is mapping this reference database in more metagenomes and metatranscriptomes so we can increase the training data set, and why not also link it to in situ optical measurements. I just forgot to include it in this slide, but I think it's something very important to further refine this workflow. And then as a recommendation, again, so the transcript ratios are good proxies of phytoplankton physiology, so it can provide EBVs as well that can be linked to remote sensing metrics. And we have to improve this in situ observation, particularly in underrepresented areas of the ocean, such as the Southern Ocean. And yeah, so let's keep working together towards this direction, and bringing this kind of conference is a very good initiative to bring people with different backgrounds together to solve big problems. So thank you for attention. Happy to take any question.
Thank you. Pedro, any question? Yes, Emmanuel.
Just regarding iron limitation or any nutrient limitation, different groups of phytoplankton react differently have different needs. And to what degree do you think you represent all the phytoplankton in the water versus certain groups that are lacking those nutrients?
So the good part is that I didn't have time to include in the presentation, but we could break down by taxonomic groups. So in fact, it's a good question because it explains the difference between the flavodoxin index and the plasocyanin index. So these mechanisms are not present in all the phytoplankton groups, and the flavodoxin is a more generic strategy, while the plasocyanin replacements of cytochrome C6 are present just in some of these groups. So what we are now trying to match is how it's explaining also the biogeography of these different phytoplankton groups in the ocean. Yeah, but
because typically when they do these bottle experiments, they get certain groups to start growing fast, while other groups do not respond, or might respond even negatively. So we have to be careful about saying phytoplankton are stressed versus certain groups of phytoplankton exhibit stress.
Thank you. Yeah, important.
Okay, thank you very much.
Welcome.
So next speaker is Peter Miller from Plymouth Marine Laboratory, UK, speaking about relationship between shelf sea fronts and biodiversity studies and using Earth observation data. Thanks, Peter.
Hi, everyone. So today I'll tell you something about larger organisms in the ocean. So we're moving beyond plankton to talk about shelf sea fronts and biodiversity. So for those not familiar with ocean fronts, this is a very obvious one. It's actually a front is the boundary between different water masses, but this is the boundary between two oceans off South Africa. Why they're important? Because these are areas where there's more mixing taking place between those two water masses. That can lead to additional nutrients coming up and extra productivity, and then that is retained there for larger animals to feed on. This is important right now, especially around the UK, with all the offshore wind farms being planned and put in. So we need to know that these are being put in areas that don't affect the marine ecosystem, and ocean fronts is one way to look at the biodiversity potential. So let's this is a summary of my talk. Let's quickly tell you about the ocean front metrics we use from satellite. So we take daily sea surface temperature and detect the fronts daily, but combine them over months and then seasons to give a distribution of those boundaries in the ocean. And then, looking at larger animals now, fish, seabirds, and mammals, we're collecting a large data set of surveys, tagging experiments around the UK, and putting that into a biodiversity map. Then the idea of this project is to relate those two parameters, the fronts with the biodiversity, and tell us about that relationship. This is for the Frontwood project for the Crown Estate, and the idea is to justify including fronts in spatial planning, particularly for offshore wind farms. So here's thermal fronts. This is an annual map of where the fronts are strongest, and we've done that for 10 years worth of data. We've also looked at ocean colour in this project, so the boundaries between high chlorophyll and low chlorophyll water, it also creates a boundary, not always in the same place. But the advantage of ULCI data is that it's high resolution, 300 metres, so we can get closer to the coast to study these hotspots of biodiversity. So let's look at how the biodiversity are measured. We combine a great deal of data, as I said, and look at the sampling effort over time and also the species counts. That's done separately for birds, mammals, and fish. And then we combine those to give this multi-year index. And I saw yesterday that sometimes with this kind of data, you do have to combine over multiple years to get the spatial coverage that you need for this type of analysis. So we combined over 20 years worth of data and the sampling season, April to September. So let's link these two together now. And I thought I'd put in an equation, but I don't like equations, so let's put that away. And for those who want it, it's there in the corner. But I like visuals, and this is preliminary results only, but this map shows the probability of detecting the particular biodiversity in these areas, only incorporating the effects of fronts. So there's many other variables that we could have put in to describe where the biodiversity are, temperature, chlorophyll, sea depth, distance from colony, that kind of thing. But this is doing a reasonable job in locating the areas where there are higher biodiversity, just based on the distribution of ocean fronts. So, as I said, it's provisional results, but we're getting a reasonable relationship, quite a high variance explained. So that's encouraging to then apply this more widely. These are the parameters that we used. So we're starting to analyse which of the parameters are more important for this biodiversity. We found the thermal front persistence, how long the front is there, and also how strong a chlorophyll front is, are two of the most significant metrics. And yeah, you can do that kind of GAM analysis to explore those relationships. So now that we have a model of biodiversity around this test region, if the fronts are moving, then the biodiversity will move with it. So we need to study what controls these front locations. You can see on this previous study of basking sharks that the fronts are moving day by day. Those are the ribbons in the background. And that the biodiversity, in this case of basking sharks, move to follow those fronts. So one thing we've done is a new algorithm to characterise how much the fronts are moving. So these ribbons, the width of the ribbon gives an idea of how much the front is moving over a week. And we're combining that over a year to give a map of where the fronts are dynamic. That's still in progress. A long-term factor is the North Atlantic Oscillation that controls the weather patterns and the storms coming more from the Atlantic. And so this is slowly varying over months and years. So we've looked at how that NAO index correlates with the fronts that we see. What's interesting when we do that is we see that a positive correlation is found where there's different types of front, a salinity front with the water coming up the English Channel, and as opposed to tidal mixing fronts. So there's interesting work there, and of course this will be important for climate change, affecting the fronts as well. Shorter term variability comes from the wind, and we started to explore this. Here's the summer data. We find that, as you might expect, wind weakens the fronts at the surface because it gets blown out. And tide. So we're finding that stronger tides lead to stronger fronts in certain areas where they're tidally mixed. And then we can do this globally. So that once we've explored and got the best relationship with biodiversity, we can start applying that to other regions. And I'd be very interested to discuss collaborations and future projects on that. So future, we need to finish off this project to address how much, where and when do ocean fronts influence the marine biodiversity. And we're describing that to the UK Crown Estate to ensure that we do make an impact with this work on spatial planning and offshore wind farms. And recommendations for the committee are to accelerate the use of ocean fronts within OBIS, GBIOS and the GeoAtlas. As an efficient, although it's a partial proxy, it only tells you about some of the biodiversity, but we find it's an important indicator. And this could help set up protected areas and hence achieve the targets for 30% protection by 2030. So thanks very much.
Thanks a lot. So there are some questions. No questions.
If not, we move to the final talk before the break. Thank you, Peter. It will be given by Ludwig Wenigan from the Polytechnic University of Catalonia in Spain. And Ludwig will tell us about large-scale automated detection of humpback and grey whales in satellite imagery using deep learning for conservation monitoring of California.
Thank you. Thank you. So, good morning, everyone. I'm Ludwig Wenigan, and on behalf of my team at the Nature Conservancy and at UPC, I would like to present our work on the detection of humpback and grey whales in California. So as for the previous presentation, we are changing scale and going from the microscopic realm to megafauna in this case. So the general context of this work is a collaboration with the Nature Conservancy and the EU Ocean Project. And in both projects, what we're aiming to do is to address the main risks to which whales are confronted, which is the risk of entanglement in fishing gear and the risk of collision with ships. So the monitoring we think that the monitoring of whales using satellite imagery can provide information that we can share with relevant authorities in order to mitigate those risks. In order to carry those objectives, we analyzed submeter satellite imagery. So this is basically commercial imagery from Maxar And here we are at a different scale also. We have imagery that has, at best, a resolution of 30 centimeters, and it oscillates between 30 centimeters and, say, 60 centimeters. We analyzed data going back to 2009, which is the first WorldView 2 data, and the last data we analyzed is from March 2023. We analyzed approximately 650,000 square kilometers, and to do all of this, we had to deal with different challenges. There was heavy maritime traffic, which meant possible false positives. We have a high turbidity in California waters, so we were not really sure if we would be able to observe the whales or even make any detection at all at the beginning of the project, but this was solved. And then we also have a lot of adverse sea states in Californian waters. So those are some examples of observation and detections of gray whales in this type of imagery. So on the left, you can see two gray whales traveling in the north of California over very turbid waters. So you can see that they actually leave a trace, an imprint on the surface of the water. The second image in the middle is near the area of Long Beach, and you can see a whale maneuvering very close to a military ship. So it gives you an idea of the actual risk of collision and why it's such an important issue for the conservation of gray whales. So that's the image of in the middle. It is very dark, so we used contrast in order to show you the gray whale at the bottom right. And then on the right, with a lower resolution, you have a pod of gray whale traveling in the north of California. In that image in particular, we had over 200 gray whales that were traveling north in the month of March. And this is the second species, humpback whale. You have seen that the gray whales were fairly clear. The humpback whales are a little bit more complicated. They are darker. You can see some traits that distinguish them, such as the very bright pectoral fins. They are darker, as I just said, and then they're also a species that breaches a lot, that moves a lot, and it makes it a little bit more challenging to detect them than a gray whale. So you will see that this reflects in the way the performance of the detection algorithm comes. So we created a database to train 21 different convolutional networks to detect gray and humpback in WorldView 1, which is a panchromatic data, WorldView 2 and WorldView 3 satellite imagery. And we used an image classification approach using pre-trained image model. And the models were adapted to use all multispectral bands available. So in the best case scenario, we have 8 multispectral bands, and we want to use all of them to increase the accuracy of our detections. And we aim to classify between 5 different targets: gray whales, ships and container ships, aircrafts, background class, and non-whale detectables, which are things that can be confounded with whales, but are not. So we wanted to challenge a little bit the model. This is a representation of the performance which we obtained for the detection of both species. In blue, you have the performance for gray whale detection models, and in red, the performance for humpback whale detection models. So on the x-axis, you have the false positive rate, and on the y-axis, the recall. And ideally, an ideal detector would be on the top left-hand corner. So you can see that the gray whale models are typically much higher than the humpback whale detection models, which correlate with what we know about behavior. We have a pretty good accuracy, almost 90% of recall in the best case scenario for the gray whales. So this is a bubble chart, and the position of the bubble, of course, relates to recall and false positive rate, and then the size of the bubble refers to the accuracy of the model. And we deploy the best models all along the coast of California. So we have detections going a little bit north of the border between California and Oregon and all the way down to the US-Mexico borders. When you zoom in, we got very interesting information about the distribution of the whales, and we got information concerning the way they travel, how far away they travel from the coast, which we did not have before. We also were able to calculate the density, and so this is a representation of the density normalized by the survey effort. So you have a very big variability of the density, and high densities are typically in red or dark red, and those are really areas that we want to put emphasis on and share the data with the authorities so that we know that those are sensitive areas that need to be protected. So just in a nutshell, the type of results that we obtain, we had gray whale detections covering the entire coast of California. We found 3,500 gray whale and 1,500 humpback whales, and opportunistic detections of blue and fin whales. The gray whale migration occurred more offshore than we expected, and the offshore migration routes through the Channel Islands feature more detection than the coastal route. So this challenged data that had been obtained about 20 years ago and that had not been updated, so this was great information. We had high densities in some areas, going up to 9.27 whales per square kilometers, and this is because this is really their migration area, and so at times we have really high densities. The median distance to the continental coast was found to be approximately 5.86 kilometers, and we had some peak values going all the way to about 57 kilometers. And as another interesting finding was that we found that there was an extended presence of humpback whales all the way through November, and there is a ship management campaign that typically stops in October, assuming that humpback whales are not present anymore. So this type of data is a way to tell the authorities that there's a possibility to extend this mandate a little bit further in time. And so regarding current and future work, we are currently deploying the algorithm towards the southern range breeding grounds of gray whales in Baja California, Mexico. And we have also started to deploy the algorithm towards the northern range summer grounds, which are located in Alaska. And we hope to have a complete vision of the northern Pacific coast. We continue the application of this method to other species, such as sperm whales, fin whales, right whale, and we aim to build a multi-species observatory. If you want to get more information about building this observatory and work on multiple species, you can take a look at the Ocean Project page. And let me go back. We have also transmitted to the International Whaling Commission how the use of satellite imagery enabled us to point that some French subantarctic islands were being recolonized by some species of right whales. And so this was very valuable information provided by Earth observation data in that case. And as far as recommendations, I just wanted to put the focus on one.
Less than 1 minute.
Just 1 minute? That's perfect. And which is to facilitate open access to large data sets of sub-meter optical imagery, particularly for endangered species, so that multiple research groups and people working on machine learning can work on those subjects. Thank you for your attention.
Thank you, and we have time for a quick question. Yes? I think we can take only one, sorry.
Thank you very much for the nice presentation. As we are about to start some exercise very similar on wells in another offshore protected area, it seems interesting to see it. And I was wondering two things, is that how you check that your data from the satellite is truth with the field data observation in case that you are missing a lot of observation, because we see detections, mapping, and provide recommendations. And I was wondering if you have double-checked that this data is with field observations from boats or aircrafts. And the other question that I have is, because we are discussing with the marine traffic, is also on the speedboats. How you check, or what is your things on your thoughts regarding the collision and the velocity for the boats to be able to avoid the collisions on that part? Thank you very much.
Thank you. So let me think. We, regarding crossing the data with in situ measurement, we did not do it. This is a very big database, but every data point that you have observed was actually verified. So we only plotted verified detections and not those that were unlikely. But yeah, this is a great idea, and we should do it. But this is a long-term project, I guess, to make this cross-reference. And then regarding the velocity, there are different programs that are implemented on the U.S. East Coast and West Coast. On the U.S. East Coast, it's for the right whales. There's a speed management program that has very clear guidelines for this. And in the Channel Island Sanctuary, there's a vessel speed reduction program that has guidelines for this.
Okay, thank you very much again.
Thank you.
So this actually ends our marine ecosystem session, and I would like really to thank all the speakers for the excellent talks. So thank you, all of them. So it is now time for coffee. It is served in the big hall. And we will reconvene here for the coastal ecosystem session. I would say that we'll start sharp at 12:10. So thank you very much. vais avoir du mal à prononcer ton nom.
Tout le monde a le problème pour prononcer maintenant.
Alors, attends. Tu es...
Je pense que le sixième.
Luba. Alors, Luba Rechitnik, c'est bien Luba?
Merci beaucoup.
Ukrainian.
Nice to meet you too.
You're Ukrainian, but you were?
I am
In
Canadian.
Canada.
I'm a Canadian Ukrainian.
Oh, okay.
Yeah.
Okay,
Hence my very Ukrainian name.
Yeah. And in Canada, where is it at?
The Hackett Institute is in British Columbia.
British
Columbia.
Okay. Thank you so much.
No, it's because someone that I used to, she's actually, she's native, but she's
You're ready? Okay, I think it's time to start again so that we are not too late for lunch. This is already a late lunch that you will have. So welcome to the second session of the day on coastal ecosystems. And here is my co-chair.
Hello. Sorry, apologies.
Thank you, Victor. I will start then by recording all the speakers again that you have 10 minutes sharp. So we will inform you after when you have 2 minutes left so that we have time for one question. And we will start with Beth Davis from University of Nantes in France. And he will be talking about a full map of European intertidal seagrass.
Hello. Right. So like Marilyn said, I'm going to be talking about intertidal seagrasses, and the title is A Full Map of European Intertidal Seagrasses. However, the paper that it's based on is actually now entitled An Initial Map of European Intertidal Seagrasses, because, you know, with all these products, there's a lot of inaccuracies, uncertainties that go forward, and that's very evident in this project as well. So this work is based off of a few different projects, but initially the BICOM project that Victor talked about yesterday, and then developing the model framework that we created in that research into a more European-wide product. So a little bit of background in intertidal seagrasses. They're a little bit more overlooked compared with their subtidal cousins. But at the same time, they can harbour really high biodiversity, can provide lots of services, and actually quite interesting ecologically due to their interface between the terrestrial and the marine environment, and the diurnal or daily changes that can happen in the area. But they're generally poorly described, or at least poorly understood, in the vast areas that they can occur at. Mostly because they're hard to access, they're quite often in areas that are quite dangerous to access on foot, and therefore remote sensing is becoming more and more frequent and prevalent in their monitoring. So, like I say, the project started off in the BICOM project, where we developed a model for detecting intertidal seagrasses in Europe. So this was a model called Ice Cream, so the intertidal classification of Europe, categorizing reflectance of emerged areas of marine vegetation with Sentinel-2. So we've published a couple of papers last summer that looked into these
this model and used it for certain sites. So looking at phenology of intertidal seagrass, how the extent of this habitat changes through the years in different sites, but then also how that changes across the whole of Europe, the scale of Europe, specific sites. But also, again, looking at how that phenology may change over time. So we're using the Sentinel-2 archive. We can't go too far back in the past, but hopefully as more data becomes available, we can start to do a little bit more long-term surveys with it. But the work I'm really going to talk about today is how we've tried to then apply the ice creams at a more continuous method, or a more consistent method across Europe. So not looking at sites that we know there are seagrasses, but actually just looking at the whole intertidal area of Europe itself. And so to do that, we use some cloud computing methods, basically taking a stack of all of the Sentinel-2 time collection and masking it for the intertidal area that we're interested in, but also looking at areas that aren't covered by cloud. So this is using some current products such as Murraytel's intertidal mask, and then also some different cloud masking techniques. And then created a stack of as many images as we can. Then from this stack, we were able to create a quality mosaic. So we took top 5 intertidal vegetation maps of Europe, so taking all of the Sentinel-2 bands where the NDVI was positive, so the vegetation was relatively positive, within these intertidal and cloud-free, we hope, areas. And then from these top 5 vegetation composites, we were then able to apply the ice creams model, so being able to create basically habitat classifications at the 10-meter resolution of Sentinel-2 and have 5 maps of this vegetation classification across the whole of the intertidal area of Europe. And then once we've got this classification, our aim was to look at seagrasses, so we picked out the seagrass class itself when it was the mode class across the 5 classifications. So as long as there was 3 out of the 5 composites that was classified as seagrass, we took that as a seagrass pixel, or at least the maximum extent, and then we can convert the NDVI of those pixels into some form of cover and create intertidal seagrass maps of potentially the max potential of intertidal seagrass across Europe. So that's our workflow. And then from this, we were able to summarize and be able to assess patterns over the different scales of Europe. So here we looked at latitude, which previously we also looked at the phenology across Europe, but we were looking very much at the sites, and this is more of a continuous assessment of phenology of intertidal seagrass. So this is looking at when the pixels were at their peak of NDVI or vegetation, when was that happening and how that changed with latitude, and actually finding that, similar to the previous work, there's quite a linear or continuous pattern of this seasonality becoming later in the year. So going from sort of August time in the northern latitudes to maybe towards November, even perhaps December in some of the more southern European sites. But then also we wanted to look at how intertidal areas were covered with seagrass, or what they were covered with. And then when they were covered with seagrass, what was the, I'm going to say density, it's not a true density, it's what were the cover classes within that seagrass area. So on the left, in the figure A, we just look at how much or what proportion of the intertidal area was covered by seagrass, regardless of what density or what cover that seagrass was. And we found it was quite consistent pattern that it became more cover of the intertidal area in Europe was covered by seagrass than in the north. This is also related to the fact there's higher areas of intertidal area in the north of Europe, so you can go into the more details when the paper's published. But then once we actually do have the seagrass, we also found patterns of higher density or higher cover seagrasses appearing further south in Europe. So for example, the 90 to 100% seagrass cover in the south of Europe was more prevalent, whereas in the north of Europe, the more prevalent classes were the 50 to 60% seagrass cover areas. And again, a lot of these patterns, not fully linear, but were fairly consistent across this latitudinal gradient. And so just to summarize, the gift did work. That's brilliant. Okay. To summarize, this is currently a paper under review with remote sensing of environment. And we're hoping to, with this paper, we're going to provide the data layer that we use to actually do these analyses. But not only the data itself, we also wanted to create some sort of user interface that you can interact with the data if you have no experience with GIS type techniques. So using Google Earth Engine for this, but maybe looking to progress to something a little bit less dependent on Google in the future, giving both the seagrass cover but also the uncertainty from our model, the timing of the pixels themselves, so day of the year, which is on the video now, and then also what year that pixel came from. So able to look at some more regional elements of maybe phenology or when the highest vegetation occurred. Then, so going into the future, what we would really be wanting to do is improve ice creams as a model itself, so improving the training data, but also actually looking at some of the products that we've used to be able to mask the intertidal area. We were getting a lot of misclassification. I think in this site, actually, we've got some salt marsh that's being classified as seagrass, and actually that's to do with the fact that we classed intertidal area as something that was fully covered at full tide, where salt marsh isn't fully covered most of the time, but we were getting some misclassification there, or misinclusion within our mask, that if we could improve those masks, that would make our actual output a lot more accurate at the end. And then, if we can, again, it's more data, but trying to apply to the global scale, but that would need a lot more validation outside of the European sphere, and then we're coming into more different species occurring that would be potential issues. Cool. Thank you very much.
Thank you. 1 minute left, so 1 or 2 questions, depending. Yeah, Masira?
Hi, thank you. Bede? Bede. Hi, Bede. It's Masira from University of Victoria in Canada. So we're doing something similar for British Columbia, a full U-grass map for in the west coast of Canada. And the approach that we're doing is using phenology for the classification, U-grass phenology and the other macroalgae phenologies as well. Is it, because you talk about phenology, but I was not clear if the phenology was used for separating the different...
So actually that was one of the first sort of tasks we went to, was trying to describe the phenology of the intertidal seagrasses, because we were seeing a lot of papers, at least either not talking about phenology in intertidal seagrasses or saying it was fairly consistent across the whole of Europe, and we didn't think that was true. And so one of the things we didn't want to include was some of the biases that could perhaps be brought in from using one phenology for the whole geographical range of the same species. And actually that was, so with the thing that we're seeing, and like you say, the seagrasses have different phenologies in different latitudes, but also I imagine different species would also have different phenological patterns.
But does it, so did you use the reflectance, the phenology / reflectance signal in the classification? That's what I was wondering.
So we just used raw classified pixels as it's a neural network model that the papers go into it a little bit more in detail, but we didn't actually have any of the temporal element in any of these models. We were just using the Sentinel-2 reflectance, standardized reflectance, or NDVI or NDWI for the classification itself, so that we weren't sort of perhaps bringing in any potential bias or assumptions in that sense, even though there are assumptions and bias in the other direction. But
Okay, thank you. Presentation will be given by Benoît Béguet from IC in France, and he will present an innovative approach for remote sensing methods and sensors benchmarking prior to blue carbon ecosystem monitoring at large scale.
Thanks, Magellan. Hi, everyone. Very happy to be there and to present a work. I'm not talking about thematic. We're more talking about method and benchmarking methods prior to blue carbon ecosystem monitoring at large scale. Obviously, this work is in the context of the ESA Blue Carbon project, a very challenging remote sensing project aiming to map mangroves. Christophe's going to talk about it maybe later, and the IRD, seagrass, and salt marshes. A very challenging remote sensing project. And to me, remote sensing starts always from local knowledge of habitats, and in that case, carbon ground estimation. And very high resolution remote sensing has shown a great potential. We saw the great presentation of Tuya just before. And it's also a resolution that offers an expert a direct vision, interpretation of nature as descriptive Earth observation. When you think about Earth observation, you think about algorithm, automatism, but just looking at an image is already Earth observation. So we start from ground knowledge, with ground people knowing their places, and they need to look at very high resolution images. What we aim is to first map the habitat extents. We saw on the presentation of Bidi that just mapping the extent is already very difficult. And I want to do this by testing the robustness of different precision levels, which is to me typologies. Once this is well done, we can try to estimate biomass for those habitats, and then obviously derive carbon maps from ground estimetric, allometric relation, which is not my job. And all of this should be applied operationally at large scale, okay? Challenge. So, what happens when you use very high resolution design reference data applied to high resolution images like Sentinel-2? Take a look at this. And obviously, that's my starting point, because this is what I want to avoid, right? Data not consistent to work at larger scale, too precise data working with drone or having small quadra on the ground. And if you use it with a wider resolution, you do something like this, very not consistent. No problem, it's an error, it's a mistake. I'm pretty sure everybody does it, but don't want to say it. Learning is doing mistakes, doing errors, understanding errors, and improving. This is what a machine learning algorithm does, and this is what we do when we want to train and progress. So we need something to avoid this. We need a standardization protocol to harmonize reference data for both habitat mapping task and biomass carbon modeling task. It's the main subject of the week, I guess. It was a long discussion yesterday afternoon about this. We all need this. I want to stay positive. I'm not going in the scientific retention of data and availability. Okay. We are working with various labs, with different approaches, different habits, very different, same habitat, different scales, different level of precision, and sometimes different units or metric systems. How to integrate field measurement and their spatial ranges for good correspondence with remote sensing data. I think it's the hardest point. And obviously, as a methodologist, I want to test, I want to benchmark sensor, remote sensing feature, explore temporality, machine learning algorithm for both classification and regression task. And I want to do this with consistency. So we want to upscale method, going from very high resolution knowledge and ground knowledge to large scale. We need to measure and understand the loss of information and control the uncertainties snowball. So I make a first proposition, a simple one. I want to force people to work with a fixed hat method based on an hexagrid, 20 meters, with like 1,000 meters square. It looks like it could be good for most of habitat we study. Obviously, at that scale, you're not going at the species level, but more habitat level. And this fixed hat method allows harmonization of reference data coming from different sources, ground, photo interpretation, existing map. This fixed spatial information integration will allow me to compare strictly sensors, Sentinel-2, planet, spot, Pleiad, remote sensing features, monotemporal, multitemporal. I could work, which is the most important for me, at pixel level, object level, or in the deep learning era, at patches level, okay? Reference data should be patches. And so, yeah, I want to test various machine learning. And I want to measure the information loss between very high resolution and high resolution before going large scale. And obviously, at the end, the object is to monitor changing and how to monitor changes with a consistent methodology. So here you have just a fast example of a Pleiad image, some ground measurement, a quite simple typology that people could photo-interpretate. So it's kind of one level we're looking for. And you got the hexagrid mesh, I-force, and you have some labelized proposition there. Looking at what's inside a small hexagrid, for Pleiad you have texture, you have information. For Sentinel-2 you have at least 3 by 3 pixels, which is the minimum to take into account. Don't look for pixel, look for patches. Because your pixel, you never know where is it exactly, right? And I forces the team to fill on the grid this kind of a table, and all information won't be available everywhere. No problem. The method will allow us to work at different level of precision. Just fill me some hexamesh like this. And when things going like this, oh, magic happens. No magic, just hard work. But hey, if everybody work hard in the same standardized protocol, things start to going well, right? And we have very, very consistent map between Pleiad and Sentinel-2 in this case. Obviously, I could be lazy and stay with a simple random forest approach. Maybe it's better, but as a methodologist, I want to test more things, segnets, conv-trad, and what could be very good is mixture of it. Like you have deep learning architecture, which allows to go on the very spatial resolution with a very high resolution. And if you have a dense time series, you will have a part of the model which is going to look at temporal patterns, which is phenology for our vegetation. So there are model mixed like this, hard to develop, but I wanted to have a hard scheme that allows me to test everything. Now I get it. And for biomass estimation, same protocol. Obviously, what everybody does the first thing is fit a model with all your data, which is just huge, awful overfitting on the left. And then you go working on feature selection. Why not random forest permutation importance or something else? And then you're going to the real world, like working on cross-validation. And yes, life is hard and painful. This is reality. So I could test many algorithms. Just a joke on this case, deep learning is not always the best solution. If you can do something with a fast, simple, random tree, don't go deep. And be parsimonious. And after this, I want to walk around my data prior to upscaling, so we can predict. No, the method is okay. I will go fast. Biomass prediction at pixel level. Okay, why not? But why not at hexagrid level? Just to test it, because if I work at hexagrid level, I will be able to compare Sentinel-2 prediction and Pleiad prediction. And we know Sentinel-2 will underestimate low values of biomass. This is what you get in this test data. And why not estimate correction factors before going large scale? I don't know why not, but I have a hard scheme that allows me to test this. And obviously, we're going to work on change detection, monitoring the evolution of habitats, and how to monitor evolution of an habitat without a fixed spatial condition. So you understand this. I just want a very controlled method to understand what I do. And so I propose this standardized approach to my colleague. Experimentation is going large scale right now in Canada, in Mediterranean, on different ecosystem, blue carbon ecosystem. To homogenize the construction of a reference database for habitat mapping, biomass estimation. I want to benchmark remote sensing sensors and features, machine learning methods, and obviously control the loss of information between very high resolution and high resolution. Monitor changes. Okay. I think it's all for me. Don't hesitate to ask me questions. And yes, many French guys working on Arcachon run. So it's just a temporary result there with all habitats, seagrass, salt marshes, and so on. Thank you.
Thank you. A super quick question, if there is one, because we are... Okay, no question. So we thank you.
How does this
Bye-bye.
Compared to the work that he is doing.
We need to work together to understand.
Okay. Because it's for entire Europe, and Europe, yeah, but they do something, but many people do the same thing.
Thank you.
Okay, so we're still with the blue carbon theme, and the next one is by Elodie Blanchard from UMR Espace Dev IRD in France. So this is about mangroves, right?
Yeah.
Fantastic, go for it. No, this is not it. This is. Whenever you're ready.
Okay, I don't see. Yeah.
You
Okay.
don't see it on your screen?
No, I don't have. I have nothing in my screen.
You have a little yeah, that's, yeah.
Yeah, but.
Okay, you can see it.
Okay, I see here. Sorry. So my name is Elodie Blanchard, and today I will show you our result on improving the. The assessment of blue carbon stock of mangroves using remote sensing along the Amazon coast. So mangrove are tropical forests that are almost on the 3/4 of coastal intertropical areas. They are located in the intertidal zone between the ocean and land, and it is a coastal blue carbon. So we know that the specificity is that this ecosystem have a high carbon sequestration capacity. So we will work on carbon of mangrove. So the carbon is sequestered in the soil, below ground and above ground. And also, more of the carbon is behind the surface. There is an important part in the trees that can be seen from space, and we will use that because we will use the above ground biomass to predict the carbon, sorry. So. The aim is to improve mapping and monitoring of mangrove above ground carbon stock, over time and across areas, using Earth observation and specifically Pleiad images. So to map carbon in French Guiana in 2017, 2020, and 2023, and extrapolate to mangrove along the Amazon coast. But today I will show results on French Guiana. So the study site is the French Guiana, which is located in northern part of South America between Brazil and Suriname. And most of the coastline is occupied by mangrove forest, which have diversity of high diversity of mangrove habitats. Mangrove reaching 45 meters, 500 tons of dry matter per hectare, and more than 800 tons carbon per hectare. So this is our huge database. So we have 105 plots that were carried out between 1997 and 2025. So in each plot we measure structure parameters like tree density, stem diameter, or tree height. We also use a large database of panchromatic Pleiad images. So we have Pleiad images all along the coast at 3 dates. So 2017, 2020, and 2024. And we also used the vector of mangrove extent. So today I will present the result on these specific areas. So we will talk about canopy texture. So on these satellite images, this is an Iconos images, you can see the canopy of mangrove. And you can see that there is different pattern, different canopy texture. So there is two examples here. So with a repetition of large crown and gaps that correspond to senescent mangroves, and another texture with high density of small crown that correspond to a young mangrove with a high density of small trees. So there is a link between canopy texture, canopy structure, and forest structure that we will use. So for the canopy texture analysis, we use the photo method, so for Fourier transform textural ordination. That is a robust method that is used since a long time. So with your image, you choose window size, you're windowing the images, and Fourier spectra is calculated for all the window, and after that you work on the variability with component principal analysis, and the score of the windows along the most relevant axes, often the three first axes, are used as texture indices. And you can represent with a map by coding the value along the axis with red, green, blue, map, composite map, sorry. So we also use a model, a multiple linear regression model that will construct with the data of above ground biomass plots measuring the field that are linked to the photo indices. And we use this model to predict the above ground biomass, like you can see on the image. And the final step is that we derive the above ground, the carbon from the above ground biomass by using equation proposed by Walker in 2018. So now the results. So we start with Pleiad images, panchromatic images at the 3 years. Here you can see the vector of mangrove extent, because I would like that you note that there is an erosion. Mangrove was eroded between 2017 and 2023. And from the Pleiad images, we obtain the texture maps. Here we have resolution of 50 meters. So you see the evolution. And from these maps, we get the above ground biomass maps. So you can see on the histogram that the biomass decreases because of the mangrove erosion. And to finish, the above ground carbon stock maps. So that, okay.
2 minutes.
Okay. So we start with Pleiad images, and by calculate the above ground biomass, we pass to the carbon stocks. But for the moment, it's just the above ground carbon, not the entirely one. That will be the next step. So, but our future work, so we will map mangrove carbon stock from French Guiana to the Amazon influence coast. We need a lot of data for that. We will improve models for predicting below ground and soil carbon stocks. We also need data, but yeah, maybe Christophe will talk about that. No, okay. You see that it's important to work on annual changements because the cost of French Guiana is really dynamic, and we will test the regional potential of time series of Sentinel-2 for carbon stock mapping, because we can't always work on Pleiad images, you understand. About the key recommendations, my principal recommendation is to step up effort to collect field data. I think it's really important, and notably to improve remote sensing carbon models, to better inform about the uncertainties of global models for predicting carbon stocks. There is a lot of models that are proposed actually, and to provide more accurate data of the amount of carbon stored by ecosystem, particularly at the level of fruit and soil system. Thank you.
Thank you very much. A quick question. Masira?
I waited, right? Someone wants to have a question, please don't. I don't need to ask. I'm just curious. So I have actually, it's not even a question, maybe it's 2 comments. You know, I think it's amazing, and if you're going to do it for the entire Amazonian coast, it's going to be incredible, right? And one thing that maybe you want to consider is integrate risks associated with the regions, right? And, you know, Sargasso is going to be a big risk because the Sargasso reaching the coast, it really kills the mangroves areas, right? Because it becomes more anoxic than it is. So the integration of the Sargasso landing or getting close to the coast with your maps will be really neat. And just in case if you're not aware, there could be in the relatively near future big oil drilling in the entire coast there, right? And the oils are going to be a big risk if they get into the mangroves area, right? So I think associating those metrics with risk would be really neat.
Yes, for sure. Thank you.
So it's just a comment.
Thank you. Thank you again. And we will welcome our next speaker will be Christophe Proisy from IRD in French Guiana. And he will present space-based monitoring of mangroves for anticipatory nature-based solutions, a 3-point research agenda.
Just a few minutes, the screen is going back. No? Yes, I got it. Okay, hi everybody. Thank you, Marie-Hélène and Victor, for the invitation. So I'm Christophe Proisy, a researcher at the IRD. My colleagues and I would like to present you how we are applying the concept of nature-based solution to mangrove in French Guiana. So, nature-based solution can be seen as action to restore, manage, and protect nature in view of addressing societal challenges, while benefiting human well-being and biodiversity itself. So it's important to understand that we need to go beyond the simple action of monitoring characterizing biodiversity. We need to work with local stakeholders to co-design, co-implement, and also co-validate this new action. For the objective is to develop new sustainable local planning. So this work is a review of what we can obtain from Earth observation to support mangrove-based solution for sustainable development in the global south. So let's go back to French Guiana. You recognize the map already presented by Elodie. So we are starting a project called Magellan. There is nothing to see with the name of this room. Huh? Sorry, it's just a coincidence. Maybe a joke from you, Marilyn. So French Guiana is a pilot site for trying to develop mangrove-based solution. French Guiana is part of the longest muddy coastline in the world, 1,500 kilometers from the Amazon River to the Orinoco River, a circulation of giant mud banks to the northwest, so very high coastal instability. I don't have time to show you all the pictures, with considerable impacts on coastal zone management. Keep in mind that mangrove in French Guiana correspond more or less and 60% of the European mangrove. So we have the chance that mangrove in French Guiana are still preserved due to low human population and activities. But this is in a rapid increase. So just two illustrations. On the left, you have the fluctuation of the coastal mangrove shoreline observed with satellite data since 1950. So the x-axis corresponds to the longitude between Brazil and Suriname, about 300 kilometers. And the y-axis corresponds to the cross-shore distance, so seaward, in the seaward direction, 8 kilometers. You can see that over years, mangrove can disappear, but reappear, following the presence and the shifting of northwest shifting of mud banks. And on the right, you have a beautiful picture showing that big trees in mangroves can be funny to climb. So let's start. The first nature-based solution we want to develop in French Guiana is, let's say, it's a kind of warning system to anticipate coastal erosion and siltation, because we already see that mangrove can adapt to coastal change. So, and we have a lot of data since 1950. So this data come from visual interpretation and more sophisticated analysis of Landsat, SPOT, Sentinel-1, Sentinel-2, and Pleiad images. And we obtain very important benefits for general understanding. First, we identified the oceanic regular waves as a major driver of coastal change in French Guiana. And mud banks protect the shoreline. So we integrate all this in a new model called MangaCoast. You can find the publication in environmental modeling and software. And this model now gives us a lot of insight for nature-based solution and for local planning, because we can predict with a short term, let's say 5 years, coastal change. And now we are working with local stakeholders on annual newsletter to inform a large audience, a large public, on coastal vulnerability in French Guiana. Second example of a nature-based solution is a warning system of changes in coastal biodiversity and resources. So the idea here is diversity of mangrove. I'm always talking about mangrove in plural. It's not a green carpet. It's a diversity of mangrove habitats, and this diversity will guarantee ecological resilience. So we use this, we try to describe this using 5 time series of very high spatial resolution to provide new data on this unknown forest. So this will support national natural reserves and even provide us means for early warning of threats to mangrove, like on the recent years, we had massive attacks of caterpillars with a lot of defoliation. So, and now benefits for research are, we are starting to better describe, quantify the trophic and resource network that allows in return to plan for socio-economic scenario and even to qualify and quantify what we are trying to do in the ESA Coastal Blue Carbon project. So we will try to provide the multi-year carbon maps. And also we are trying to consolidate the carbon approach using a major project in France called Tropicos, funded by the Fair Carbon program. Last nature-based solution is a monitoring system of changes in mangrove and human habitats together. Because mangrove can provide a lot of services to urbanization, to get a sustainable urbanization, we would like to study how maps, fine-scale maps of mangrove can be integrated, can be directly integrated into urban planning decision for better health and better well-being. So services like water, phyto depuration, wind protection, air temperature reduction, and so on can be useful for next local planning. Let's see also mangrove as indicator of environmental degradation, coastal erosion, as you already see, but also upstream salinization, detrimental to concrete artificialization. So this new way of thinking led us to define new project, to submit a new project, like a detection system of emerging pandemics brought by mosquitoes. So it's time to conclude. So see the nature-based solution as an innovative and integrative approach. And I would like to demonstrate that the mangrove case study in French Guiana is quite advanced to see mangrove as powerful to anticipate change in coastal landscape, change in coastal biodiversity and resources, but also change in coastal health and well-being. For me, we need to continue to validate Earth observation methods, so through multi-scale strategy, but also by considering not only the biodiversity, but also the human habitats. They are evolving all together. And we need humility, because it's not, mangroves are complex, nature is complex, so we need field validation and a well understanding of signal physics. So it's a question of credibility with local stakeholders. And my last slide is about our recommendation. So biodiversity needs interdisciplinary research. We already say that. Second point, biodiversity needs also sound Earth observation data sensor. And for the tropics, I would like to highlight the future potential of the P-band biomass mission. But for me, overall, the overarching recommendation is to get closer to local stakeholders and the public, because Earth observation deliver facts, not opinions. It's important to be clear on this and to claim it loud and clear. So biodiversity for all of us matters, so it's important to continue delivering this message. Okay, thank you.
Thank you, Christophe. Any question apart from Messier?
No, you could.
No, you can. No? If not, we move on?
Yes.
Thank you very much.
Welcome.
So I have two announcements to make. The first one is that we are entering the 3 last talks, and then there is lunch. So we have to be a bit more efficient and be there probably the first ones because we're the closest to the canteen.
Yeah, that's a good
So,
point.
It's a good point. So I was going to propose, Marilyn, that as we get the people coming in, who the next is Luba, just start coming in. For the other ones, I was thinking to get prepared while we're going with the questions. So we get the people ready when they are going with the questions, right? And the other thing is, second announcement, to make a little break, is that we have the marine workshop in the afternoon. It's not in this building, but in the last building. I don't know if you've been before. It's a small room in the separate building. If anyone wants to go, we'll try to organize a small shuttle in the reception like 15 minutes earlier. So if you don't know where you're going, 15 minutes before the session at the reception here in the front. So are we ready to go?
I'm going to wait for the screen to come online.
That's all right.
You have a third announcement? Maybe?
I can have a third announcement. Oh yeah, we can stand up and do some relaxation because we've been sitting for a long time. So we can do some exercise. I don't know.
Please do while we fix this.
Thank you.
Yes,
We have
brava. Well done. Okay. Are we ready? Okay.
But I think you can start maybe with...
Yeah.
Okay.
So there you go.
I'll do my best, but I don't have my notes. Buon pomeriggio.
You need your notes.
I would like them, but we are short on time. So all is good.
What do you need?
I don't see my notes on the screen.
You have notes on the screen.
Neither did most of the other speakers.
I don't think they did.
No, that's okay. Hi, everyone. My name is Luba Rashidnik. I'm a researcher with the Hackeye Institute in British Columbia, and today I'm going to be presenting on our coastwide kelp mapping efforts. This is done in partnership with colleagues at the Woods Hole Oceanographic Institute and other colleagues at Hackeye. So, let me bring you closer to the ocean, a little bit away from our coastlines, but still pretty nearby. Kelp forests are found across 25% of global coastlines where they provide valuable ecological, economic, and cultural services. In a recent study by Aaron Egger et al., when comparing kelp forests to urchin barrens, they consistently found higher ecosystem services found as both increased biodiversity as well as increased species richness. In British Columbia, we have 2 dominant species of canopy-forming kelp, very charismatic, that are driven and are constantly experiencing a suite of environmental and biological drivers, which leads to naturally very high variability in these ecosystems. But because they have these large, emergent, floating canopies, this makes them amenable to a suite of different remote sensing technologies, from drones all the way to coarser resolution, satellite resolution. And one of the satellites that we do use when it comes to mapping kelp forests is the Landsat time series. This provides global coverage since 1984 at a 30-meter resolution. And there have been many studies that have demonstrated the application of using Landsat for both examining changes in kelp forests through time, looking at those drivers, but also giving us a really important historical context for when we do see new changes happening in these ecosystems. And so with that, the objective of this work is to produce the first full map of kelp forests in British Columbia using the Landsat time series. And we also want to leverage high-resolution aerial imagery that the Hackeye Institute collects in order to obtain species-level distributions. So we do this using a fixed-wing aircraft that collects 4-band, 10-centimeter resolution imagery.
Wow.
We classify using an AI, which saves me a lot of time. And so how we do this is using methods described in Tom Bell's 2023 paper. And for the sake of time, I will not go through the details here, but understand that we can do this. Land masking and intertidal masking is extremely important, as well as the final step, which requires a manual review of the data sets. And in order to look at a time series of kelp canopy extent, the maximum extent that we detect in a single year, we require that pixels meet a certain threshold for the number of detections in a given year. And this is due to variations in the number of Landsat sensors we might have in any given year and how that's increased through time, as well as looking at in certain years that maybe were more or less impacted by cloud cover. We can also use these data sets to create a map of persistence. So how many times has a given pixel been detected as kelp over this time period? As you can imagine, manually classifying 25,000 kilometers of coastline is quite the process, and that would make British Columbia the third largest country in Europe after Norway and Greenland. So for today, I will be showing you hot off the presses results we have for both the north coast and central coast of British Columbia. So along the north coast, we have a region that is dominated by what were once historically incredibly large kelp forests. Here visualized in the persistence map, where we see higher detections of kelp in the bluer or greener colors. And we can look at that overlap with what kelp looked like back in 2023, represented by these red pixels. So really a contraction, a decrease in that total kelp area that we detected that year. When we look at that time series data, we can look at it in two ways here, both at the tile level represented by that yellow polygon, as well as this outer coast area. And we can see a few things in this graph. A, that we're missing some data because of those really conservative requirements to be a part of this time series, but that we also get a sense of what those past trends look like, that we see lots of years where there was high amounts of kelp, low amounts of kelp. And actually, when we look at the statistical analysis of this, we did find a statistically significant decrease in kelp when we looked at those outer coast areas compared to that whole tile value. And you can see from 2016 onwards, really this anomaly of a sustained, persistent pattern of low kelp. And this is likely due to following a blob event, what we call like this massive warm water, as well as other probably interacting drivers as well. Heading down further south to the central coast, where the Hackeye Institute has its our field station, and really a beautiful part of the world. We can, by first starting with our time series, we see something really different. We see not statistically significant, but a lot of variability and actually increasing over time. And this is a place where we've been doing that fixed wing aerial survey imagery. And so a few things to show you here. We can look at that tile level again, so that yellow polygon. We also have this beautiful offshore island group called the Goose Group, where we do our fixed wing surveys. The data you can see in the larger inset is the persistent maps, so these kind of embayments, these rocky reefs. And then looking in that smaller inset, we see kelp classified as giant kelp in yellow, bull kelp in blue, and the Landsat data with the red pixels. And so you can kind of immediately see some of the trends that we might be detecting with Landsat that were actually, that the aerial data are telling us that we might be missing due to certain reasons. When we compare kelp canopy derived from fixed wing data over the same time period of Landsat, we can start to see that we have some really good agreement in the total canopy kelp mapped over time, but also helping us understand where we might see deviations in patterns of extent over time. And what the aerial data provide us that the Landsat data don't is understanding like how much kelp and where is it by a species level. What we can see here is that over time, giant kelp is our dominant species of kelp in this area, with the bull kelp being present in a lower proportion, and that also translates to what we detect as well. So kind of understanding the limitations of Landsat when it comes to mapping these plants that are actually typically much smaller, and their biomass is present in typically much sparser ways, in particular on this part of the coast. And so with that, I hope you understand our main takeaways of the first look at using Landsat for mapping kelp at a BC-wide context, and that we can use these data to see regional differences in trends in kelp canopy extent, and how it's really important to leverage this high-resolution imagery to validate Landsat data products, as well as understanding species-level distribution and trends. So our future work here is focused on completing this time series, so hopefully within the next 6 months we'll have that full view done. And this is actually a piece of work that's nested in a larger collaborative effort with folks like Mysira at the UVic Spectral Lab to examine drivers of change of kelp across British Columbia. We also know that we need higher spatial and temporal resolution, and so we're currently working on a proposal with the Canadian Space Agency to build on pipelines to classify both Sentinel and PlanetDove data, which is also work that we've been doing with MyCERA. And importantly, too, there's the KelpWatch platform, which provides a really cool way for visualizing these data and allowing others to both discover, see what kelp looks like, and download those data directly as CSVs. So really leveraging those data and making them much more accessible. And so finally, kind of my recommendations, kelp mapping, and especially remote sensing, has benefited from just such a wonderful community of practice. We have an annual meeting where we nerd out and talk about this. We've been able to produce publications as well as more publicly friendly guidebooks. And really recommendations too in this space involve learning and engaging about conversations in EO data and indigenous data sovereignty, while also thinking about how we mobilize those data sets and tools, while also taking that into account. So a thing I'm thinking about a lot this week. Thank you.
Thank you very much.
Thank you very much. We have time for one question. Do you need your notes?
No, really
no.
not.
Thank you.
What you wanted for? Anyway, one question? No? Next speaker? Getting ready? Yeah? Thank you very much. And fantastic.
Thank you. So the next speaker will be Hajar Sadel Imani.
Yeah.
Hopefully it's not too bad pronunciation. And she will be speaking on spatial-temporal evaluation and hyperspectral modeling of microphytobenthos cross-primary productivity in France estuarine environment.
Yeah, so hello everyone, my name is Hajar. And I will present the spatiotemporal evaluation and hyperspectral modeling of microphytobenthos cross-primary production in estuarine environments. So this presentation is in the context of Rewrite project. Rewrite is an European project that aims on the restoration of coastal ecosystems and the biodiversity changes. So, as we know, coastal ecosystems are generally underestimated in the global carbon budget, specifically for mudflat. However, one of the key contributors is microphytobenthos. So what's microphytobenthos? Microphytobenthos is defined as a microscopic organisms living in the surface of mudflat that storage carbon by the photosynthesis activity, and the amount of carbon stored is what we call gross primary production. So the amount of carbon that is stored in the coastal ecosystem is estimated to be around 500 megaton of carbon by year. So to improve this understanding, my research tried to link between carbon fluxes measurements and hyperspectral remote sensing techniques to create maps and temporal scenarios that aim to evaluate how these ecosystems contribute to the global carbon budget. So yeah, currently I'm working in 3 estuary environments in the western part of France that are Loire, Seine, and Canche. So how we measure the carbon fluxes in these ecosystems, specifically for microphytobenthos, we are using the infrared gas analyzer instrumentation that is based in the infrared light to measure the CO2 concentration, using eventually the light chambers and the dark one. The light one is for the photosynthesis and the dark one for the respiration process. And then we can compute the net ecosystem change from the slow variation of CO2 concentration. So then the GPP is related to the net ecosystem change, and we derive the GPP from the net ecosystem change at the light condition and dark. So we just observed that the values of net ecosystem change in these ecosystems at the light conditions are much higher than the dark. So we can say that microphytobenthos is highly productive. So, yeah, that's my little toy. I'm using I'm using a hyperspectral camera to capture imagery at the footprint of a chamber. So here we have the specific hyperspectral signature of microphytobenthos. So for field campaigns, I'm working on 5 sites across 3 estuary environments in France. Seasons are autumn and summer, 24. We are caring about GPP measurements, temperature, and light conditions. And so far we have data, hyperspectral data for 2 stations. Yeah, I just tried to summarize the method that we are still in progress of developing and improving. So from the CO2 fluxes, measurements, and climatic variables, we derive GPP and PAR and temperature that are spatiotemporal evaluated. Then for the remote sensing data, from the hyperspectral imagery, we enhance this imagery by the noise correction. And from the footprint of chambers, we compute some sensitive hyperspectral indices related to biomass and light conditions. And all these variables are used as input for developing a multiple linear regression model to be able to compute GPP as the hyperspectral index, PAR, and temperature. So here some hyperspectral sensitive indices that has been developed, the classical one and the VI, but what is more interesting is the microphytobenthos index and the light use efficiency microphytobenthos. Yeah, here we have the two-dimensional statistical analysis of GPP in function of environmental factors. So for PAR and GPP, we just realized that high GPP values are related to high PAR values, but not always, because sometimes it's related to temperature too. So for the temperature, higher GPP are at higher Maxwell temperature, but lower one are observed in the autumn season. So here we have the hyperspectral indices that has been computed from the footprint of chambers and evaluated with GPP. So here we have primarily statistical analysis from some sites. We just realized that GPP can be related to environmental factors as light condition and temperature, but not only, also with hyperspectral indices, specifically for the light use efficiency microphytobenthos. So the spatial variability of GPP can be explained to the variation in sediment type or the nutrient availability. The temporal patterns can be also related to temperature and light conditions, and for the hyperspectral indices, but we are still trying to discover other indices that are more related to microphytobenthos. We can say until now that the light use efficiency microphytobenthos is strongly related with the GPP. So for our future work, that's true that we are working in the footprint of a chamber with high spatial resolution to be able to develop an accurate hyperspectral model. But the idea is to upscale this model using satellites, hyperspectral data,
not only for the hyperspectral, but also for the thermal data, for temperature, and maybe for power from other sources as the eddy covariance, used also for the measurements of the concentration of CO2 across the European sites. Okay. So, as key recommendations, I would like to say that high resolution, we need to couple between high resolution, hyperspectral high resolution and thermal data to expand the network of eddy covariance. Maybe we don't have enough eddy covariance data to be able to upscale the model at larger scale, especially for coastal ecosystems, and to support long-term of field work. So, yeah, thank you.
Thank you very much. Some question to Aja?
Yes.
No question. So then we'll move to the last talk. I think.
Yes.
Thank you very much.
Thank you. So, is this Simon? Yes, Simon Ori, you're the last one of the session. Yes, you are. There you go. So, welcome and thank you for coming to talk to us about this very interesting topic of pigment composition, right?
Yep,
Well
the
done.
pigment, but you will see. You will see.
I'm very, very excited. Very excited. Almost as excited as the lunch.
Okay, let's go. So thank you everyone to be there. So I'm Simon Wary, a PhD student at the Nantes University in France, and I'm here to present you the work I've been doing for the past several months about the impact of marine and atmospheric heat wave on the intertidal seagrasses through the eye of an experiment, spectroradiometry, and using also satellite. So a bit of context, this work, the idea of this work started back in June 2022 when we were on the field for the ESA-funded BICOM project in Portugal. So we were looking for seagrasses on one of our sites, and we have seen some strange, we can say, behavior of the seagrasses, where some part of the meadow were totally discolored, as you can see here on the right part of your screen. So you have some part of the meadow that is totally white, and some other part of the meadow that have become totally dark, and that also has been observed by some colleague of ours in a site in Quiberon in France, where you can see huge patches here of the meadow that has been totally darkened following an event of extreme sharp increase in the water temperature and the air temperature at that site. So that's a bit the context in which this study takes place. So what is the literature speaking about the impact of heat wave on seagrasses? So most of the studies are working on subtidal seagrasses, and they have shown that there is a huge impact of course, heat wave on those species, notably in terms of physiology. It's impacting a lot the physiology of those algae, of those macrophytes, sorry. But none of these studies is speaking about intertidal seagrasses, meaning seagrasses that develop between low and high tide. And none of them is speaking about the impact on the reflectance on those seagrasses. So that's what we are going to do. So the hypothesis of this work is that heat wave alter the spectral reflectance of those Terranoltei seagrasses, and that this change can be detected using remote sensing technique. So to do that, we're gonna start with an experiment where we're gonna stress some sample of those Terranoltei in the lab and to measure reflectance on it during a long time. Based on the finding of this experiment, we will build a new spectral radiometric index specially designed to detect the thermal stress on seagrasses. And then we're gonna apply those findings to a satellite-based scenario around a heat wave that occurred in September 2021 in Quiberon, France. So first, I'm going to develop the method of the experiment. So for the experiment, we use two intertidal chambers that you can see here in a picture. And this chamber allows us to control the temperature of the air inside of the chamber at low tide, the temperature of the water. We can control the tidal cycle, and we can control the photoperiod and the light intensity also. And inside of those chamber, we can measure spectroradiometric spectra, hyperspectral spectra, using an ISD with a fiber optic. And inside of the chamber, we're gonna put our sample, so a sample of Zostera nulltei seagrasses, and we're gonna expose those samples to two different scenarios. One will be the control, where seagrass will be exposed to normal temperature of the season, and the other sample will be exposed to a treatment scenario, where seagrass will be exposed to heat wave-like temperature of both the air and the water. And for the satellite, so we will study a heat wave event that occurred in Quiberon, France, on the 11th of September 2021. So around that event, we have three images, one before the event, one during the event, and another one 1 month after the event. And also, around a few days after the event, we have some colleagues that have performed a field trip where they took a lot of pictures, quadrant pictures of the seagrasses, and to assess the status of the seagrasses after the event. So let's talk about the results. You have here the hyperspectral signature of the experiment, with on the left the control spectra and on the right the treatment spectra. So you can see that for the control, the spectra are really stable a long time, and the spectral shape is very characteristic to a seagrass spectra with really high reflectance in the green, a strong absorption in the red, and a high infrared plateau. When we look at the treatment spectra, we can see that for the first day of experiment, the spectra are looking like the one of the control. But for day 2 and day 3 of exposure, we can see that there is a strong decrease. We can say that the reflectance is decreasing a lot in the green part of the spectra, but also in the infrared part of the spectra. So based on that observation, we decided to build a new index that will take into account those characteristics to detect the darkening in seagrasses. Basically, what we have done, we have drawn an interpolated line between the reflectance at 560 nanometer and 842 nanometer, and we have compared that interpolation with the real reflectance value. And when the interpolated line is higher than the real reflectance, then our index is positive, meaning that there is darkening in the seagrass. And when the reflectance is higher, then the index is negative, meaning that there is no darkening of seagrasses. So in our experiments, we tested a lot of spectral indices, and all of them show a decrease along time of their value. But the most sensitive of them all was the one we built, the SHSI for seagrass heat shock index. It was the most sensitive index we tested because it's changes of more than 420% at the end of the experiment. If we speak about the satellite images now, so you have here the image of our site, where you have the image on the left that is before the heatwave event, and the image on the right that is during the event. So just visually, you can see that there is darkening of some pixel. And the points are representing the field campaign that we perform. And we categorize the picture we took on the field in two categories. Impacted seagrasses in golden point, and unimpacted seagrasses in green point. So when we look at the spectral shape of those two categories of points before the heat wave, we can see that there is no significant difference in the spectral shape. They are both looking like normal seagrass spectra. But when we look at the same points after or during the heat wave, we can see that the golden impacted points are showing a spectral signature that is really similar to what we saw during our experiment, with a strong decrease in the reflectance of the green and in the infrared. So when we apply the SHSI to those satellite images, we can see that on the 6th of September, so during the event, the SHSI is increasing a lot, reaching more than 100% of difference. whereas the ones that have not been impacted by the heat wave are showing almost no differences. And 1 month after, on the 8th of October, we can see that the SHSI values are back to their normal value. When we look at the map, you can see that there is a spatial pattern. Like, it's not random the way the seagrass have darkened. And this can be explained by the emergent time of those pixels. So we have the bathymetry, so we are able to compute for each pixel how long this pixel is exposed to the air, and we can see that there is a threshold among which the seagrass are darkening a lot, around 13 hours a day. So let's discuss about those results. So we have shown that, 2 minutes, it's going to be all right. So we have shown that seagrass are impacted by heat wave and that they are showing a significant spectral signature, making it possible to detect those changes using remote sensing technique with the index we built, the SHSI. And so the index has been designed to be used with most of the special space mission used today, but also we will be able to use it with future missions such as Sentinel-2 Next Generation and Landsat Next. So this is really important to be able to map and to detect the impact of heat wave on seagrasses, because there is, the literature has shown that there is a rapid escalation globally of the heat wave frequency, intensity, and duration. And this trend can be also observed locally in my site of Quiberon, where you have here, for example, the frequency and the cumulative intensity of the heat wave since 40 years ago. And that can, the fact that the frequency and the intensity is increasing a long time might impact the resilience of those ecosystems of seagrasses to heat wave. That's it. Thanks a lot.
So thank you very much. We have time for one question.
Yes.
Yes.
Hi, thanks so much for that. My question was, did you see a recovery in the thermal index? Like, looking at your map from the Sentinel-derived, it looked like in the later time period, yeah,
exactly, there is a recovery, but there is a recovery for sure, because you can see here on the middle image that it's really yellow where it's darkened.
Yeah.
And 1 month after, it's blue again.
Okay.
So there is a recovery, because it's intertidal seagrasses, so the leaves are growing really fast.
Mm-hmm.
When they burn,
okay,
they
so you think there's
detach.
turnover and like new leaves that have grown?
The leaf detach, and then new leaf is growing.
Do you have a sense, based on this work or work that you might do, on how much thermal stress is too much before you see
No,
like,
that's
plant
something
death?
I want to do for the future experiments.
Awesome, very
because
exciting.
Here it's just like really a binary experiment that I've done, really normal temperature and really high temperature, but I want to do a gradient of temperature to test which temperature is okay, which is not.
Okay, go on. Yeah, go on, Masira.
Thank you, Marie-Hélène. Thank you, Vito. I was wondering, spectrally speaking, do you guys have epiphytes on Ugrass?
Yes.
So how confused it gets spectrally speaking?
For sure it is. There is epiphytes that are contributed to the spectral signature of the spectra, and notably in the infrared. Then how much it contributes to the overall spectral signature, I don't know. And also I don't know how much it's impacted also by the heat wave. I don't know if epiphytes are impacted by the heat wave. What I see is just like the combination of the two.
Okay. Well, thank you very much. Thank you, Simon.
You're welcome.
Very nice
talk.
Thank you. Thank you, everyone. Thank you, everyone, for staying around. I will. I just wanted to repeat the invitation for the workshop this afternoon. It will start at 3. Of those who may get lost to get there, we may find someone to take you from there in reception at 10 to 3 in reception. If you don't know where you're going, 10 to 3 in reception. And have a nice lunch. Marie-Hélène, I'll let Marie-Hélène close.
No, thank you all, and enjoy your meal, and thank you to my co-chair.
Thanks.
And to all the speakers. Yeah, indeed. indeed, indeed. Very nice to meet you, and I hope I was not. I tried. We need people like that.